What happened in our phase 1 trial is we've been treating 14 patients in the trial, and 8 patients doing the dose escalation and 6 patients at the highest dose. So far we have 3 response, 1 at the dose escalation and the 2 at the response at the highest dose. And for that we have 1 cervical cancer patient and 1 esophageal cancer patient, and lastly 1 urothelial cancer patient. And those patients have been doing well, and most importantly, since this is the first CD4 T cell therapy, and we are glad to see this phase 1 study has been safe, and we see some early response, and we hope that in the phase 2 trial we can ask the question whether that the response will extend to many different types of cancer. There are 2 types of T cell. One is CD4 T cell, or people call it as helper T cell. Another type of T cell called killer T cell, CD8 T cell. So usually people think that the CD8 T cell is in charge of the killing, so to eliminate tumor cell, and CD4 T cell just provide the help. But today we want to ask the question whether CD4 T cell alone not just provide help, but also itself can in charge of the killing. For scientists that both in the clinical study and also preclinical study, that we haven't found the specific mechanism that induce that. So far we think maybe 1 cytokine is called interferon gamma may play an important role in that, but other than that, it's still very limited. So I think that the success of this clinical trial will help scientists and stimulate scientists to doing more study on the CD4 T cell, because as we mentioned before, that we have a bias that CD4 T cell only provide help, but it may do much more than that. So about the MAGE-A3, it's one of the member of the antigen group. It's called cancer gemini antigen. So those type of antigen is they expressed by doing the fetal development, but when people grow up to the adult, those expression have been lost, and so the only few places that those antigen will be expressed. One is in the germline cells, and the other one is the tumor cell. So that's why we choose that as a target, because since it doesn't express in the normal cell, so we avoid the dangerous to target the T cell against the normal cells. We don't think so. Yeah, we don't think they have anything to do with the autoimmunity in normal human, because the normal human normal tissue, they don't express MAGE-A3. So far, our major direction is to enroll patients from different cancer specimens, as long as they are MAGE-A3 positive. And as we mentioned, that we focus on a few that's highly MAGE-A3 positive, such as melanoma, esophageal cancer, urothelial cancer, and cervical cancer. And so we'll try to ask the question whether this treatment will help those patients to have significant response, and most important, the long-term survival for those patients. And once we have that, that's the first step. And the second step we'll do is to find a different way to enhance the strength of the CD4 T cell, and then we may initiate different trials to test those hypotheses. So for the entire cancer immunotherapy field, that one of the first important things is choose the target. So we think the MAGE-A3 is a good target, and second consideration is what type of approach we want to use. Either use a CAR or use a TCR, and then before that, by default, people think just use a CD8 T cell, either use CAR or TCR. But now we provide a new direction that people would like to try, which is the CD4 T cell. The dose that we use to define depends on the toxicity that we observe. So the design of the trial is we start with 10 million cells with one patient. If we observe any dose-limiting toxicity, we'll stay with that dose and treat for 6 patients. And since we did not observe any toxicity, so that's why we move out to the highest dose and treat for 6 patients to complete the study. So that's a major purpose for the phase 1 study, is for safety. And then for the phase 2, then we'll study the effectiveness and the efficacy for this trial. So at this time, then we'll start ask questions and just use the highest cell dose to do that.